Good afternoon to all. Welcome to e course on plant tissue culture organized by Vinayak Vignan Mahavidyalay, Nandgaon, Khandweshwar, Amravati. First of all, I should thank organizer for giving me opportunity to share my experiences on plant tissue culture area, and thankful to Alka Bhise ma'am, principal of Vinayak Vignan Mahavidyalay. I'm also thankful to Dr. Suchita Kudke, Head Botany Department. I'm also thankful to Dr. Gajendra Singh Pachlore and Dr. Shilananda Hiverle sir for helping me during the preparation of this particular platform. I'm also thankful to Dr. Varsha Hutke ma'am, who is working at GVISH Amravati, for suggesting and co-working on this particular e-course. I will be talking in two languages during this entire course, Hindi and English, because I think we have reasonably large number of participants who are joining on this e-course from various parts of our country. And therefore, I think I am comfortable talking in both the languages, Hindi as well as English. I'm also thankful to all students, teachers, and participants for joining this particular e-course, because I think this particular e-course will be a really good course for many beginners who wish to develop a plant tissue culture lab. So, with that, I'm really excited to begin on this particular session. So, today's session is a basic concept of plant tissue culture. I am Dheeraj Naik, working at Department of Botany, Bajaj College of Science, Wardha. My email ID is naikdheeraj at gmail.com. So you can reach me on this email if you want to share some of your experiences, or if you want to discuss any topic. I'll be ready to share my experiences too. So let us begin on this particular topic. So today I'm going to discuss on what is plant tissue culture, what are the advantages of tissue culture over the intact plant, or what are the advantages of applications of tissue cultures. Then we are going to discuss on general terminology which are used in plant tissue culture area. We will see what various landmarks and the history and in fact the discovery made by different scientists in area of plant tissue culture. Then we will discuss on pathways of plant regeneration and types of tissue culture. We will also discuss on some of the key parameters which require to manipulate different manipulation in plant tissue culture activities, and we will discuss on basic requirement of plant tissue cultures. We will also discuss what are the steps which are used to develop tissue culture based plant. So these are some of the topics which we are going to discuss today. So let us begin. What is plant tissue culture? Plant tissue culture is basically a collection of various techniques which are used to maintain or growth of plants, plant cell tissues as well as organ under sterile conditions. So it is also called as a aseptic culture of plant cells and tissues under control conditions to lead to a regeneration of organs and whole plants. So you can take say any type of a tissue or any kind of a organ, and when you are able to get a complete shoot formation or root formation or somat embryo formation, that type of process is called as aseptic culture of plant tissues. It is also called as a growth and maintenance of plant cells, tissues, and organs and whole plants under in vitro conditions. So inside the glass, inside the sterilized glass vessels, when you develop mass multiplied tissue culture plants, it is a plant tissue culture process. Now, what are the advantages of tissue culture over intact plants? So what are the applications of these tissue culture rays over intact plants? First thing is, one of the very commonly used method of plant tissue culture is micropropagation, and micropropagation is basically in vitro clonal propagation by using axillary shoots or meristem cultures. So when you use micropropagation for any plant species, you are able to develop relatively large number of uniform individuals from that particular starting material, and therefore you are able to basically conserve rare and endangered species, or you can mass multiply any novel variety of say roses, banana, or any kind of a horticulture plants like gerbera, anthurium, geranium, petunia. So there are many horticulture plants and floriculture plants which requires to be developed for a large scale and which can be possible with the help of tissue culture practices. Second advantage of tissue culture application is one can able to develop transgenic plants by using plant tissue culture process. For example, let's say we have developed a BT cotton, so you require a regeneration protocol. You have to have a complete organ formation, shoot formation as well as root formation from those leaf discs which were used to co-cultivate with the Agrobacterium tumefaciens, which is one of the natural genetic engineering bacteria which help us to transfer a tDNA. So most of the transgenic plants which are developed so far have developed with the help of a very strong reproducible regeneration system which is developed with the help of tissue culture process. A third important technology which is developed with the help of tissue culture is anther culture, where we are able to develop haploid plants by using anther cultures. And one of the greatest advantage of these haploid plants is one can develop dihaploid plants which can shorten the entire breeding process, because to develop pure lines you have to do a back crossing with your parental line to develop a homozygous lines, and these homozygous lines for one particular traits or those varieties which can be developed with the help of anther cultures. Anther culture derived basically plantlets. The next advantage of this tissue culture is basically when you want to do a distant hybridization. Obviously, when you go for normal pollination and fertilization, most of the time, even if the pollination works, fertilization fail. And even if fertilization works, there is a reasonable amount of abnormality in the seeds, as well as the percentage of hybrid formation is relatively very low. And therefore, in that case, what is the best alternative is you basically isolate the protoplast from these two distant varieties or distant species and fuse these protoplasts with the help of different types of electrical or chemical or physical method to develop a somatic hybrids. And once you get those somatic hybrids or those fused protoplasts, you can basically regenerate a callus from those fused protoplasts, and that callus can give rise to a shoot formation as well as root formation or complete plantlet, and that plant is going to be having characters from one species and characters from other species as well. So this is one of the greatest advantage to develop, and that has been used extensively in tomatoes and many other Solanaceous members. Similarly, since I have raised a word called callus culture is basically unorganized mass of cells, which is basically a de-differentiation. Any tissue like petiole, leaf lamina, axillary bud, stem, any root tissues, when they end up developing callus, that callus can be trimmed into a small pieces, and you can make a liquid culture from this callus culture, which is basically single cell culture or suspension culture. And one of the greatest advantage of this suspension culture is you can grow them just like similar to the bacteria, and therefore one can in mass multiply the growth of these plant cells in a 500 ml bioreactor or 100 ml bioreactor. So by having such a large amount of plant cells, one can use it for continuous extraction of secondary metabolites and many other valuable products. So that is one of the greatest advantage for many medicinal plants and those plants which gives us various valuable products in our day-to-day life. Another advantage of these cell suspension cultures is when you have so many cell suspensions, so many cells, these cells sometimes undergo some of the epigenetic changes and end up producing permanent variations, and these are called as a somaclonal variation. And these somaclonal variation basically helps to create different types of variants, and one can use a one particular trait, let's say for example any secondary metabolites which is important, and we can have a high yielding cell lines developed from this kind of a screening procedure. So these are the advantage of tissue culture raised plants over the intact plants. Let us see first general terminology used in plant tissue culture. I have already used various terminologies and various terms in just in last 2 minutes in discussion. The first terminology which is very commonly used is adventitious shoot formation or root formation or de novo shoot formation. These are the terminology which is very commonly used in micropropagation and organogenesis. So what is it? It is basically the shoot formation or root formation. Adventitious shoots are those shoots that arises from any location on the plant except in the axil of the leaf at the node. They can form on the stem internodes, leaves, roots, or callus. What does this really mean? That there are three places where there is a pre-existing meristem. One is a apical shoot, another is say a root, and the third is the axillary meristem. Now, these three meristems are called as a pre-existing meristem. Okay, so these pre-existing meristem, whenever you give a cytokinine treatment to these tissues in the form of media, they just proliferate and develop a elongated shoot. But apart from these axillary shoots or pre-existing shoot, when they proliferate, there is again a new meristem formation which happens in the in between the cambial tissue, which is active enough in a stem or in other regions. That new shoot formation or new meristem formation gives rise to many shoots, and since these shoots arises from not from pre-existing meristem, it is called as a de novo meristem formation, de novo shoot formation, or adventitious shoot formation. Now the next terminology which is we are going to use quite often is explant. Now what is explant? Explant is any plant part which is taken from the plant body, the tissue which is taken from its original sites, and then it is transferred to the nutrient media for growth and maintenance and for development of various protocols. For example, axillary bud, apical bud, internode or internodal stem or internodal explant, petiole, leaf lamina. Here I really mean by leaf disc, pedicellate stem. So inflorescence, any part of the inflorescence, which is either could be a stem, a peduncle, pieces of that, we can use it. We can use sepals, petals, anthers, unfertilized ovule, fertilized ovule, seeds, some part of the fruits, roots, hypocotyle, cotyledon, gynoecium, embryos, and endospermic tissues. So all these different plant part are called as explant. Next terminology is very important terminology, is called differentiation. Now what happens when the meristem develops? Meristem undergo mitotic cell division, and after the cells undergo again elongation, and after elongation they follow a differentiation process. So either that particular tissue determines to form a leaf tissue, or it determines to form a stem tissue. So that kind of a specialized development of cells and tissues is called as a differentiation. So cells undergo differentiation, that technically means they form a specific tissues or organ with specific function. Next terminology is de-differentiation. What is de-differentiation? Is that this is very strongly associated with the callus, since callus is widely recognized as a proliferating mass of de-differentiated cells. So whenever any explant, whether it's a leaf, shoot, stem, root, or any inflorescence which is highly specialized organ undergo developmental phases to form a callus, that process is called as a de-differentiation, and it is strictly reversal of basically differentiation. Then there is one more terminology which is called redifferentiation. And now redifferentiation is basically that whenever a callus, which is unorganized mass of cells, which is undifferentiated mass of cells, these differentiated mass of cells develop various meristematic clusters within the callus tissue, and these meristematic clusters have competence to form a shoot formation or root formation or embryo development, and that process is called as a redifferentiation. Now the next terminology we will see is regeneration. Regeneration is basically any kind of a overall organogenesis or any kind of a morphogenetic response which is either developed in the form of complete embryo formation, or it is either in the form of shoot formation, root formation, or any kind of other organ formation. For example, there is a microtuberization also happens. There is a development of flower development also. In vitro flowering is also possible. So all kinds of these responses are called as a regeneration. One more terminology which is very commonly used in plant tissue culture is subculture, or sometimes it is also called as a passage. Now sometimes there is a slight difference between passage and subculture. Passage is, for example, let's say you have inoculated a banana seromeristem or a meristematic tissue. Now, whenever you use any kind of a somewhat woody tissue, woody explant, and inoculate on a media, in the first few weeks, they basically release lots of phenolics, and because of which the media become colored, and it become sometimes that phenolics become phototoxic to the explant, and explant eventually die or desiccate. So in that case, what happened? You basically take the same explant and put it on a fresh media immediately after 3 4 days. Or sometimes you take certain shoots which are not large enough in a size, they are just producing two or three, and if you want to put it on a fresh media, it is called passage. But subculture is, let's say you have inoculated axillary bud and you got say 20 shoots, and now these 20 multiple shoots you want to subdivide. So you basically chop them or separate them from one another. So for example, let's say from 20 shoots you are producing two shoots. So technically you are producing 10 different shoot clusters, and now you will inoculate these shoot clusters on a fresh media, and that process is called as a subculture. So similarly, if you have a callus blob of tissue and that you want to subculture on the media, you basically chop into smaller pieces, and those smaller pieces, when you transfer on a fresh culture media, that is called subculture. Aseptic technique, basically aseptic condition, and aseptic technique is mean the same, that the procedure where we want to prevent the introduction of fungi, bacteria, viruses, and other microorganisms into culture, and that particular process is called as a aseptic technique. So that is about the various terminology which are used in uh, plant tissue culture. Okay, now let us begin with the history of plant tissue culture studies. C. Haberland is the first person who has developed isolated plant cells and in vitro condition on artificial nutrient media, and he is considered as the first person who has started a tissue culture, plant tissue culture. After 1902, when in 1922, Robbins and Cote are the first one who has cultured the isolated roots for a very short period of time, where the real organogenesis uh, concept began. Then in 1934, P.R. White is the again a very well-known plant tissue culturist, which has shown a demonstration of indefinite cultures of tomato roots for a very long period of time. So he could prepare these tomato roots. He can develop a root cultures, and these root cultures were able to subculture over a long period of time, and that was shown by P.R. White. And then in 1939, Gothard and Nobecoat were the first one who have developed a very long-term culture of callus from cambium tissues, which were isolated from carrot tissues. And therefore, at that time, carrot was one of the very favorite model to study tissue culture practices. Later on in 1939, almost in late 40s, P.R. White again has developed a callus culture of tomato from tumor tissues of interspecific hybrids of Nicotiana glaucum and Nicotiana longstaffi. So, as I said, White was one of the very famous plant tissue culturist at that time. Then at later on 1941, Van Overbeek showed a nutritional discovered the nutritional value of liquid endosperm of coconut for cultures of isolate carrot embryos. So embryogenesis was achieved by using the liquid endosperm, which we commonly called as coconut water or coconut milk, and that coconut milk has a very relatively large concentrations of auxins, which are competent enough to induce embryos in carrot. So in 1948, Kaplan and Stewarts again used coconut milk along with the 2,4-D for proliferation of cultured of carrot and potato tissues. So 2,4-D is again 2,4-dichlorophenolic acid, which is again very in early days it was used as a herbicide, but later on it was found that that has auxin-like activity, and therefore it is used in development of different tissue culture protocols. Then in 1953, Moore was the first one which has inoculated the callus culture in liquid media to develop a cell suspension culture, which he could able to subculture again, and that was the first time Moore could show that we can develop a single cell culture or suspension culture. Later on in 1955, Miller and Skoog identified a cytokinin, kinetin. Kinetin is again a synthetic cytokinin, and which was shown to be a potent cell division factor. So later on, in 1957, Skoog is again a very famous plant tissue culturist, and Miller, they hypothesized that shoot and root initiation in culture callus is regulated by the proportion of auxins and cytokinin in culture media. So what they have found out, that if you have a very high concentration of cytokinin with no auxin, then that callus will give rise to a lot of shoot formation, adventitious shoot formation. Whereas when you have very high concentration of auxin and no cytokinin, in that condition you will have a root formation, adventitious root formation from the callus. And if you have intermediate concentration of cytokinin and auxin, not high, not low, but intermediate concentration of both the cytokinin as well as auxin, then that will give rise to just callus proliferation. So it was clearly a cytokinin auxin ratio which determines the shoot formation and root formation. So this hypothesis was proved by Skoog and Miller. Then in 1960, Koking was the first one who has used enzymatic based isolation and culture of protoplast, and that was the beginning of protoplast culture. In 1960, G. Morrell was the first one who has developed shoot apex culture. Now, this particular technique is reasonably interesting. For example, you basically isolate meristem, and since the meristem, a very prominent meristem, do not have any vascular connections, and therefore they are generally free of viruses, and therefore one can prepare virus-free plants by using shoot apical, what is called as a meristem culture or shoot apex culture. And that was the discoveries which was made by Morrell. Then a famous Indian plant biologist, Sipra Guha Mukherjee and S.C. Maheshwari, they have cultured anthers and pollens of the Datura stramonium and produced haploid embryos, androgenic embryos, and these embryos were, when they did the cytological analysis of this, they found that they are haploid plants. So this was one of the most prominent discovery which was then used in genetic improvement of many crop plants. And we should be proud that, okay, some of the Indian famous plant biologists have contributed such an immense observation at that time. Then Melcher produces somatic hybrids from attached plasmids vectors into a naked protoplast. So it was basically a transformation of a plasmid vector into a naked protoplast, and that was the transformation protocol. Then in 1983, Chilton was the first one who has developed a transgenic tobacco from single cell transformation. So those are some of the major discoveries in plant tissue culture area. Now we'll see the pathways of plant regeneration. What I mean by pathways of plant generation is that how cells decide to undergo different types of tissue formation. Now, typically, when we inoculate for the first time, we inoculate the X plant under aseptic condition, which are either, as I said, axillary buds, apical buds, internodal explants, seeds. And these tissues, when we inoculate them on a different media, either Murashige, Skoog media, and there are different 20 to 30 different types of media which are formulated by different scientists. Murashige and Skoog are the two scientists who has developed, and that's why we call it as a MS media. So in this media, when you add cytokinins or when you add auxins, these tissues, they undergo de-differentiation, and that de-differentiated tissue forms a callus. And when the callus undergo redifferentiation, it forms a organogenesis or embryogenesis, and that is generally called as a indirect organogenesis or indirect regeneration. Now, if you look at the other tissues like axillary bud or leaf disc, when they have certain competent cells, and these competent cells undergo to form induction because of the auxins or cytokinin concentration, they become a determined cells, and these determined cells either form a shoot formation or root formation or embryo formation, and that kind of a process is called as a direct regeneration or direct organogenesis. Similarly, if you take these tissues and isolate a protoplast, and from a protoplast, when you culture these protoplasts, they will have a regeneration of cell wall, and once they have a cell wall, that single cell will give rise to few other cells, and then there is a cluster of cells. These cluster of cells give rise to callus, and this callus will again give rise to shoot formation. And so the entire pathway of plant regeneration depends upon the de-differentiation process, the induction process, which is also called as a competence, and then the formation of organ, which is also again called as a differentiation process. Now let us see the different types of tissue cultures. The first prominent one is micropropagation. The next one is called as organogenesis or regeneration, which either could be a shoot regeneration if there is a shoot formation, or root regeneration if there is a root formation, and that is also called as a root organogenesis. And the third one is any of these explant can give rise to embryos, which are, since the tissue is somatic, they are called as a somatic embryogenesis. And once these embryo achieve a cotyledonary stage, they are ready enough to germinate and form a complete plantlet. So that is a somatic embryogenesis and germination to form a complete plantlet. Next to that is anther culture. As I already told, Dr. Sipra Guha Mukherjee and Dr. S.C. Maheshwari, these are two famous plant biologists, have used anther cultures, and similarly pollen cultures. So either you inoculate the intact anther, or you basically tease the anthers and take the pollens and put it on a media, and they will give rise to a haploid embryos. So these are called as androgenic embryos, since they are from the anther tissues. Then one can use, after the fertilized embryo, you have a lot of endosperm surrounded by that particular embryo, and that endosperm is triploid in nature. So if you grow this endosperm or inoculate that endosperm, it will give rise to a callus formation, or sometimes it can give rise to even shoot formation or root formation. So if you get those shoots, there is a likely chances that cytologically they are triploid in nature. And there are various advantages of triploids, right? The triploids are basically seedless. So if you can produce the seedless plants by using an endosperm culture. Another important technique is protoplast culture and formation of somatic hybrids. As I already told you, that when you have two distinct species and you want to hybridize them, that distant hybridization sometimes doesn't work by normal pollination or hybridization technique. And in that case, if you're able to isolate the protoplast from leaf tissues, culture them, and proliferate them to form say callus, and if that callus gives rise to shoot formation, but of course during the culture, before the culture formation, if you fuse two protoplasts, then that fused protoplast will have the characteristics from genomic characteristics from the one of the variety as well as from other varieties, and that will give a completely new hybrid, and that hybrid is called as somatic hybrids. Again, callus and suspension cultures. Callus basically helps to it is an undifferentiated mass of cells. It is a unorganized mass of cells. When you unorganized mass of cells, you chop into small pieces, put it on a liquid media, that will form a cell suspension. Just like a bacterial culture, it will form a plant cell suspension culture. And this particular technique is very much useful for enhance determination or enhancement of secondary metabolites and many valuable products. So those are some of the types of techniques, types of tissue cultures, and I think we will discuss in more detail about entire those technique. Even Varsha ma'am will discuss on some of those technique in more detail. So I will not go in depth of those techniques. I will go ahead and talk about the key parameters for manipulation of plant tissue culture. The first one is nutrient media. So there are various types of nutrient media which are right now available in the market, and they are available in the literature as well. Then, as I told you, that you have to use different types of explants, and these explants could be a in vitro germinated seedling, it is a seed, it is axillary bud, apical bud, petiole, leaf lamina, anther, sepal, petal, and so on and so forth. There are plenty of explants available on a plant body. Then again, the growth environment. So once you inoculate this explant, you have to incubate this explant to a growth environment, and those growth environment are generally have either into a growth room, which should have, or growth incubator, and they should have a light source available. They should have a temperature control, which is either you can do with the help of air conditioning, and some amount of humidity should be present in that culture growth environment. So if you modulate these three factors, then there is a chances that you will have a very successful culturing of that particular plant species, and where you can see the growth and development of that entire plant. Now let us see some of the key parameters for manipulation of plant tissue culture. The first key parameter is culture media ingredients. And if you look at this diagram carefully, what you can see that the culture media is basically subcategorized or sub categorized into uh, few components. First component is called as a macro element, where potassium, phosphorus, calcium, magnesium, and sulfur is the important. And therefore, if you look at the Morashiki scoop media, you will see potassium nitrate, ammonium nitrate, potassium dihydrogen phosphate, calcium chloride, magnesium sulfate. So these are the some of the macro elements which are absolutely essential for growth and development. Similarly, in a Morashiki scoop media, you will also see micronutrient which has a iron source, manganese, zinc, boron, copper, molybdenum, and cobalt, and they have a reasonably low concentration as compared to what the macro element concentrations are. Apart from these macro element and micro element, you will also require a iron source which is provided in the form of this iron sulfate, and along with that you will have sodium salt of ethylene diamine tetraacetic acid, which basically helps to chelate this iron reasonably well in a aqueous solution. So apart from this macro element and micro element and iron source, you will also have a vitamin, that is thiamine, nicotinic acid, pyridoxine, HCl or pyridoxine hydrochloric acid, and then there is a one more sugar alcohol which we add in the media, which is either called as a mesoinositol or myoinositol. So apart from these three ingredients, macro element, micro element, and vitamin, you will also see the sucrose or the sugar in the form of sucrose, or you can add others as well. So there are various reports where other sugars like glucose, fructose, and many others have added in the form of 20 to 40 gram per liter, which is 3% or 2% to 4%. uh, Apart from these, you can also add plant growth regulators. These plant growth regulators, you prepare the stocks of plant growth regulators. For example, in cytokinin, the natural cytokinin is zeatin, but the other synthetic cytokinins are basically benzylaminopurin, kinetin, and few others. Uh, Apart from these cytokinins, you can also add growth regulators like auxins, gibberellic acids, and few others. And these growth regulators generally either get dissolved into sodium hydroxide solution or in a few drops of ethanol, and then you once they dissolve, you make the final volume with the water. So those are the stocks, and everywhere you have to make the stock solutions of macro element, micro element, iron source, vitamin source. Generally, we don't make a stock of mesoinositol because it gets easily contaminated with the fungi if you reuse it again and again. Plant growth regulators also need to be dissolved in ethanol or in sodium hydroxide. And then for semi-solid media, you add agar. Agar is one of the very cheap and easily available polysaccharides, which is act as a gelling agent. Sometimes some people have tried agarose as well. Agarose is a bit expensive, and therefore people prefer to use agar. Gel-right or phytagel is a pure form of agar, and therefore whenever you want to show the root development, since the agar has a semi-solid nature and it is whitish in color, many times the complete root development, the observation of root development is not possible. And therefore in that case, if you really want to see from where the roots are emerging from, how many roots are coming, in what time they are elongating, all those kinds of developmental phases of roots and few other features, the gel-right is the best option because it is a transparent agar. The entire tube, though it is a semi-solid, it is transparent in nature. So in support system, sometimes instead of using this agarose or gel-right, in a liquid media, you can add some paper cubes inside if your cultures proliferate much faster in liquid media instead of semi-solid media. So that's about the tissue culture ingredients. Now, there are certain formulations of these culture media which are commercially available. They, of course, they differ in a salt concentration and the presence and absence of salt as well. So there are some salts which are available in, say, Murashik Skoog media, but the same salt may not be utilized by Lloyd and Macon, which is called as woody plant media. This woody plant media is extensively used for tissue cultures of forest trees and woody shrubs. But for example, gambos is have heavily used for root cultures of some of the solanaceous member. And you can see that there are n number of tissue culture media which are formulated by various scientists. Lisman and Skoog, that is also extensively used in tissue cultures of gymnospermic plant. Lloyd and Macron is very common for tissue cultures of forest trees, and few others are available. Murashige Skoog is one of the highly cited, commonly used tissue culture media in most of the plant species, whether it's a horticulture, floriculture, medicinal plants, crop species, and forest species. Now, if you look at the essential elements of these ingredients, as you can see that the macro element like nitrogen, potassium, calcium, magnesium, phosphorus, sulfur, they basically are important and they are building blocks of proteins, nucleic acid, coenzymes, and therefore they are involved in a plant cell biochemistry, right? Whereas the chlorine, iron, manganese, these are involved in development of components of cytochrome, enzyme cofactors, and chlorine and few others like iron source is required photosynthesis because chlorophyll has iron clusters and few other things. Cobalt, copper, zinc, and molybdenum, they are also act as enzyme cofactors, which are the enzymes which are involved in chlorophyll biosynthesis, in nitrogen metabolism, and in photosynthetic electron transport chain as well. So all these macro elements and micro elements are absolutely essential components of plant cell growth. Now, apart from those ingredients, the nutrient ingredients, plant tissue culture media also uses plant growth regulators. For example, one of the commonly used plant growth regulators are indole acetic acid, indole butyric acid. Indole butyric acid is a synthetic auxin, and which basically is more significant as far as its performance is concerned as compared to indole 3-acetic acid. So indole butyric acid is in fact the low concentration of indole butyric acid provides reasonably large number of root induction in many plant species. 2,4-D is a 2,4-dichlorophenoxyacetic acid. Although it is auxin, it is extensively used for callus production. 2,4,5-D is also used for callus production, and there are few other synthetic auxins which are written here. For example, NOA, MCPA, dicamba, picloram. These are also used for sometimes for Schur induction, for root induction, and a few other properties of tissue cultures. Apart from this auxin, there are cytokinins are also extensively used. In fact, BAP or BA, which is 6-benzylamminopurine, and benzyladenine, these two cytokinins are extensively used in micropropagation of many plant species because these particular cytokinin, which are the synthetic cytokinin, proliferate the axillary bud and apical buds, and therefore their use is very extensive in micropropagation protocols of many plant species. Although apart from these two, kinetin and zeatin is also used in combinations with the BAP to get a higher percentage of shoot proliferation. One of the interesting cytokinin is thiodiazuron, which has auxin-like activity as well as cytokinin-like activity, and this particular cytokinin is extensively used in recent years for development of somatic embryos, for shoot proliferation, for root proliferation, for callus production. One of my colleagues who used to use this thiodiazuron for micropropagation of carnations, and he used to literally get 80 to 100 adventitious shoots per explant or per single culture vessel, and he used to get massive multiplication rate for carnaceous species by using this particular. Thiodiazuron is a very promising plant growth regulator which has shown a very high multiplication rate in micropropagation. Now we'll see some of the basic requirements of plant tissue culture. Obviously, you require plant material or mother plant, and this mother plant has to be elite, or it has to have some important characters for which you require mass multiplication of that particular species. Obviously, whenever you decide to use that particular mother plant, it should be healthy, disease free, so that then the chances of surface sterilize and developing aseptic culture is reasonably easy. Apart from plant material, obviously you will require lots of equipments and glasswares, which we'll be discussing in detail in the next session. But basically, in equipments you require laminar hood, autoclave, oven, microwave, pH meter, and few others. Apart from these equipment and glasses, you also require aseptic conditions and basically clean lab environment, a laminar hood. So these two conditions, clean lab environment, is in fact one of the most critical one to maintain your tissue culture lab. I mean, you can initiate the tissue culture lab in any lab, but if you want to maintain a very good quality tissue culture lab, your lab has to be clean. It has to be cleaned by various types of lab cleaners which are available. Phenyl, or you can use any kind of a Dettol, for example. You can clean the lab and equipments and the surfaces of your benches with those detergents or with those cleaning ingredients. Next one is washing and storage facility. Although for beginners, even if you have a small size storage facility or washing basin, that is still fine, because when you are a beginner, you literally want to establish your lab. So this washing facility doesn't have to be a really large enough. But generally in commercial tissue culture labs, they have a separate washing room and they have separate storage facilities. Then they also have a media preparation area. Sometimes they have media preparation rooms, sterilization rooms, and transfer rooms as well. So these rooms are, they are separated from each other. But as I said, those people who are beginner or just want to establish a tissue culture lab in their college or in their institution, even that is a single lab, that doesn't going to impact the tissue culture development, because you can prepare a media on one simple bench. You can sterilize with the help of small autoclave, which is maybe a 10 liters of it. And even if you have a small size, say laminar hood, which where a single person can comfortably sit and do inoculation of say 50 test tubes or 20 test tubes, that kind of a laminar hood is still enough to begin for plant tissue culture work. Then culture rooms, if you have a separate culture room, well and good. But if you don't have it, if you have a rack and in that room you have an air cooler or air conditioner, that's a perfect lab for you. What you have to do is you have to have those culture racks with the fluorescent tube lights, and those fluorescent tube lights should have a sensor or a small a timer where you can keep the lights for 16 hours or 12 hours of light on, or 8 to 10 hours of off. I mean, complete darkness should be there. So that can be customized by using local vendors or even an electrician which is available in your institution, which is not that difficult. And well equipped observation area means you should able to stand there or sit there to observe your tissue culture uh, uh, test tubes and vessels comfortably, so that you can if there are any contamination, you should able to remove those contamination. And you have to see these cultures quite often. You just inoculate and incubate them and you forget about these culture racks or tissue culture vessels, then that it is not going to work. You have to have a continuous monitoring of this tissue culture uh, vessels. So that's about the requirement. Now we'll see the steps in plant tissue culture, and these are some of the most common steps in developing any kind of types of tissue culture protocol for that particular plant species. First is a selection of promising elite plant material, which we call mother plant, for collecting the explant. Second thing is the isolation of explant. You should able to isolate that particular explant reasonably. I mean, in a proper phase. Then you should able to surface sterilize that explant with the help of sodium hypochlorite solution with the 70% ethanol, or with the end any kind of a fungicide solution. And then at the end you should able to wash them with the sterile distilled water, and then you should able to inoculate that explant in a sterile or autoclave nutrient media. Now that nutrient media you can pour into test tubes, in conical flask, in magenta bottles, or in bioreactors. you can use in a you can have a small size bioreactors, or maybe a 1 liter of conical flask. You can do even inoculation in a 1 liter of conical flask if you have reasonably large amount of callus culture which is available to develop suspension culture. So all these types of vessels you can use to inoculate your explants. Then you should able to incubate these explant on that, obviously on that sterile nutrient media in a controlled incubation condition, where I've said the temperature should be 25 plus and minus 2 degrees Celsius. But in some cases you can keep even at 30 degrees Celsius. It doesn't really matter. Temperature, of course, you have to optimize that, but in many places we cannot simply optimize the temperature because we don't have a relatively large number of incubation condition. Otherwise you have to run many experiments to study how the temperature is influencing the morphogenetic response of that particular tissue culture. So if you have a limited facility having 25 plus and minus 2 degrees Celsius, with the help of air conditioning, with the help of air cooler if it is possible, then you make it. You have to have a 16 hours of light, 8 hours of dark condition. That is also depends. Sometimes some people can keep a continuous light, but keeping continuous dark won't be good, because these cultures, they do partial photosynthesis, they are partially heterotrophic, so light is absolutely important for the morphogenetic responses of these plants. And therefore relative humidity, which is basically maintained by the that particular air cooler or air conditioner, that humidity is enough for incubation of those cultures. Obviously, the next stage, once you have these cultures just proliferating from that explant, you should able to passage them into a new fresh media. Should able to subculture. If you're getting a elongated shoot, then you can chop into many pieces and again inoculate those small auxiliary bud. That is called subculturing. You can also able to, if you get elongated shoot, you should able to put those shoots on a rooting media, which could be again a morassic scoob media with the indole acetic acid or indole butyric acid, or any kind of oxygen which will give rise to a rooting response. Sometimes some people try ex vitro rooting. Ex vitro rooting is basically, once you have elongated shoots, you should directly dip them into a oxygen solution and put that shoot into some kind of a perlite plus vermiculite plus little bit of brick and sand, small miniaturized pot, and cover that particular elongated shoots with a plastic bag and maintain the humidity. Sometimes you don't have to have this in vitro rooting. You will get direct rooting on the tip of that cut portion of that elongated shoot, and sometimes this ex vitro rooting found to be acclimatizing or hardening more efficiently than the in vitro rooted plantlet. And the last stage, which is again one of the limiting stage for commercial tissue culture plant or for any kind of a tissue culture project, that acclimatization and hardening of these rooted in vitro raised plants or even germinated embryos from these most optimum condition to somewhat what we call as a harsh condition, or the conditions which are there in the real environment. So that kind of a acclimatization process from partial heterotrophic to complete heterotrophic condition, with a less humidity, with a high temperature, with a strong light intensity, sometimes during this condition plants can easily die, and therefore this particular stage could be a limiting factor for many plant tissues. The last stage is once you have acclimatized plants, hardened plants, you simply have to transfer them into open field and just verify them or study them, or see the performance of these plants under field condition with compared to seedlings or cuttings or any other vegetative propagated plants, so that you can compare whether the micropropagated plants are performing much better than the other types of plants. So that's about the steps in tissue culture, and those steps are the common steps in most different types of tissue culture. I have referred to this, and I generally recommend this book which is written by Bojwani and Razdan, Plant Tissue Culture Theory and Practices. It is been revised many times and it is printed in India. It is available in India, and this is really a Bible of plant tissue culture. At least in India, it can be considered as a Bible for the plant tissue culture area. Thank you. If you have any questions, you can drop me email or we will have a discussion at the end of this session. So thank you.